Thank you for that introduction, and thank you for joining us today. It is my pleasure to begin our webinar on achieving reproducible, publication-ready protein expression data. We all know that Western blot is a workhorse method in protein analysis. About 20,000 articles containing Western blot data are published every year. For decades, researchers have been relying on Western blot to study various aspects of protein biochemistry. Western blot is an immunoassay that provides specific detection of target proteins in complex biological samples. Since it was developed in 1979, Western blot has evolved relatively little, and the basic steps of separation, transfer, staining, and visualization have remained the same. There are three commonly encountered problems with Western blot data. First, the lengthy and manual workflow is a significant contributing factor to the unreliability of Western blot data. Almost every step introduces difficult-to-control variables that impact the final results. A second problem with Western blot data is that the data are primarily qualitative, and extracting quantitative information is difficult and often unreliable. A third major challenge and a potential blind spot for Western blot users is that Western blots are easily susceptible to misrepresentation, often motivated by misguided efforts to make the images cleaner for publication. Examples include cutting out lanes to rearrange sample order or non-uniformly changing contrast settings to enhance visualizations of weak bands. Unfortunately, misrepresentation of Western blot data is surprisingly common. An eye-opening fact is that 25% of retracted articles in the biological sciences have image problems, and that Western blot accounts for approximately half of publications with image problems. According to Retraction Watch, the biological sciences sees over 1,000 image-related retractions every year, and because Western blot is the culprit of around 50 to 60% of retracted articles with image problems, it is not unreasonable to estimate that around 500 retractions occur every year because of problematic Western blot data. Furthermore, these retractions can cause career damage, with recent high-profile instances of resignations after improper data were revealed in publications. So what if we could reimagine a better way to do protein analysis? Western blots are still considered the gold standard for protein detection and characterization, whereas ELISAs are considered the gold standard for protein quantitation. Simple Western is a relatively new technology that combines the benefits of these two methods to offer a completely novel way to analyze proteins. Simple Western is a capillary electrophoresis-based immunoassay. Let's talk about how things work in the capillary. At its core, capillaries are what enabled this advance in capability, and the foundational innovation was being able to enable capillary electrophoresis and pair it with UV-activated sample immobilization to replace the variable transfer step of traditional Western blots and allow for in-capillary immunodetection. Simple Western technology is fundamentally enabled by capillaries. The instrument starts by automatically loading the separation matrices into the capillaries. This is followed by loading of a small sample plug in each capillary. Proteins are then separated by their apparent molecular weight, like SDS page. The next step, crucial to the process, is that proteins are immobilized to the capillary wall using UV light. This allows for the removal of the inert separation materials from the capillary. Your Simple Western instrument then performs an immunoassay inside each capillary using the primary antibody of your choice, and are ready to use secondary antibodies for easy and sensitive detection of your target. The instrument images the signal from the immunoassay in each capillary and automatically analyzes the data in our Compass software. Simple Western technology uses capillaries to do an SDS-based size separation of proteins in a sample, immobilize them to the capillary wall, and then do immunodetection with primary and secondary antibodies. Looking at the evolution of Western blot, it was first invented 45 years ago And the traditional manual approach, utilizing slab gels and transferring to a PVDF membrane for immunostaining, is still used today. Semi-automated Western blotting tools started emerging around the 2000s and provided modest workflow improvements by automating portions of the workflow, such as transfer or staining. Then, in 2011, Protein Simple, a biotechnology brand, launched the first fully automated capillary-based Western blot, called Simon. 10 years ago, an improved instrument called WES combined the individual capillaries into pre-assembled cartridges and offered significant improvements in sensitivity, robustness, and ease of use. In 2018, the JESS system added multiplexing capabilities, and in 2021, a budget-friendly ABI system included a method called replex, a stripping and reprobing alternative to maximize data obtainable from each sample and run. Simple Western enables enhanced quantitation and throughput, so researchers can use Western-based readouts for applications they would never consider using a traditional Western blot for, including both upstream and downstream development and commercial stage assays. Today, Simple Western offers 3 different platforms. The ABI system offers chemiluminescent detection, and the JESS system offers chemiluminescent and fluorescent detection. Both JESS and ABI can run 25 samples in just 3 hours. The new LEO system allows runs of up to 100 samples in the same 3-hour timeframe. Simple Western is precise and gives quantitative results in under 3 hours. The platform is sample and target agnostic and improves high throughput and flexibility. It's highly reproducible with intra-assay CVs below 15%, requires minimal sample volume, and delivers exceptional sensitivity. Currently, researchers are using Simple Western in a wide variety of applications where traditional Western blots and other immunoassay technologies on the market struggle to deliver with enough speed, specificity, and quantitation, and some of those are shown on this slide. As we know, there are many steps to a Western blot, and you may even be familiar with or using products to make parts of the workflow slightly easier or better. This includes using precast gels, higher throughput electrophoresis chambers, faster transfer equipment, automated Western blot processors, or advanced imaging systems. These individual tools, or the combination of them in series, is sometimes marketed as automated Western blotting. But when you think of it, you still need to carry out each step. Simple Western technology is the only solution on the market to automate the entire Western workflow. In other words, it doesn't do parts of the process for you, it does the entire process for you from sample to data in one hands-free run. To summarize, using Simple Western is easy and mostly hands-free. Once you finish preparing your samples and reagents and loading the sample plate, Simple Western does the rest of the work for you, and you get fully analyzed data in about 3 hours. No manual interventions, no washing plates or membranes. So, what does Simple Western data look like, and why is it superior to traditional Western blot data? Compass for Simple Western is the control and data analysis application for Simple Western instruments. Compass lets you set up assays and streamlines data analysis with its intuitive interface and robust tools for automating protein quantification and annotation. Once a run is complete, the results are automatically presented in the software with several options to choose from to view and analyze your data. One way to view Simple Western data is the electropherogram, which provides detailed high-resolution and quantitative Western analysis. The electropherogram is a Cartesian plot of signal intensity by molecular weight along the capillary. The peaks and valleys of an electropherogram better illuminate isoforms, cross-reactivity, or degradation products, as opposed to crude image data of traditional Western blots. Compass automatically calculates signal height, area, percent area, width, signal-to-noise ratio, baseline values, and even absolute concentration using a standard curve. These quantitative measurements are presented in a results table along with the graph view of your data. Traditional Western blot users may be more accustomed to viewing their data as bands on a gel. Therefore, Compass also provides the option to view simple Western data in a virtual lane view. Instead of peaks on an electropherogram, in lane view, your target is presented as bands, just like an imaged membrane from traditional Western blot. Because each capillary is a self-contained assay, the lanes can be easily reordered using just a drag and drop of the mouse. This eliminates having to rerun samples in a desired order and reduces risk of improper image manipulation, such as splicing or duplication of lanes. With both electropherogram and lane view, users may annotate and export figures for publication. There's no need for additional software. Transparency is the key to quality results, and simple Western data are fully transparent. All raw data, including assay setup, run settings, and analysis settings, are captured in self-contained run files that are easily shared or uploaded to data repositories. To summarize this part of the webinar, Simple Western provides a solution to a complex and frankly outdated technique, ultimately giving Western data without the challenges of Western blotting, and elevating protein analysis to a level beyond that of traditional Western blotting. Simple Western automates the entire Western blotting workflow and provides fully quantitative results in 3 hours. In the next part of this webinar, Aino Sormunen, a doctoral researcher from the University of Eastern Finland, will present a comparison of automated and traditional Western blotting methods.
Hello, and thank you for the introduction. My name is Aino Sormunen, and I'm a doctoral researcher at the AI Virtanen Institute in the University of Eastern Finland. The reason I'm here today is that a couple years ago, me and my colleagues published an article where we compared the traditional Western blotting method to a couple of automated ones, and today I will walk you through a short summary of the work that we did. So probably most of you are familiar with the Western blotting procedure to some extent, but if you're not, then here it is in a nutshell. It is one of the most often used analytical techniques for identifying specific target proteins from complex protein samples, such as tissues or cell lysates. And this is normally done by first separating the proteins from each other by size using gel electrophoresis. Next, the proteins are transferred from the gel onto a membrane. After this, the target proteins are probed with specific antibodies, which then enables the production of an enzyme-activated chemiluminescence signal, and this signal can then be visualized to detect and quantify the proteins. Even though Western blotting is one of the most often used techniques to detect proteins, there are a lot of problems still associated with it. The process itself is very time-consuming, and it also needs large amounts of protein and therefore also sample to be able to detect the proteins in a sufficient way. This is especially problematic if the proteins you are targeting are expressed at low amounts in the tissues or cells, or if in general your samples are very precious or limited. In addition, the normalization of the target proteins can be quite unreliable sometimes, and as I'm sure you've heard in the previous talk, there are a lot of problems in reproducibility. These problems can arise because of multiple reasons, but they can include, for example, the fact that different steps of the procedure can be performed in different ways, and that different laboratories often use different reagents for the same steps. To combat these difficulties, different degrees of automation have been developed for different parts of the Western blotting process. One example of this is the semi-automated iBind Flex device. So, sample preparation, gel electrophoresis, membrane transfer, and imaging are performed exactly as they are in traditional Western blotting, but then the device can perform all of the blocking, antibody incubations, and washes using sequential lateral flow. This is what the device looks like. So basically you put your membrane here in the center, you close the lid, and then on top of the device there's another lid that opens, and here you can pipette your blocking solutions, antibody solutions, and washing solutions. You close everything and then you just let it run for 2.5 hours on its own. And after that, you can proceed as normal into the imaging steps. Another example is the simple Western Jess, that is a fully automated and capillary-based system. The system performs all of the steps from protein size separation and immunoblotting all the way up until imaging and analysis of the images. This then leaves sample preparation, like sample dilutions and antibody dilutions and other kinds of preparation, as well as pipetting to a special plate to the user. So then on to the methods and what we actually did in these experiments. So to create our samples, we used adenoviral vector plasmids that expressed 4 different sized transgenes that all included the receptor binding domain of the coronavirus SARS-CoV-2 spike protein. And as a control, we used a construct that expressed the vascular endothelial growth factor VGF. So we had 4 RBD constructs that expressed all a different sized protein, and then we have the control construct. And these were all transfected into HEC293T cells using the lipofectamine 3000 reagents. And 48 hours after the transfection, the cells were lysed and collected, and then we proceeded into the Western blot. So in case you're not familiar with the Western workflow, you can see it there on the right. And here are all the details of the reagents and such. So for protein loading, we used either 10, 2.5, or 1 micrograms of total protein per lane. For blocking the membrane, we used 5% bovine serum albumin in TBST. And here you can see the antibody information. So we had an antibody detecting the SARS-CoV-2 receptor binding domain. And then for our housekeeping gene, we had an anti-GAPDH antibody. And since both of our primary antibodies were rabbit, we had a secondary antibody that was an HRB conjugated goat anti-rabbit antibody. For detection, we used Pierce ECL Plus Western blotting substrate, and then the images were taken using the Biorad ChemiDoc imaging system. Next for the iBind, so here I've highlighted in bold the reagents that have changed compared to the traditional Western blot. So for blocking and antibody dilutions and washing, we used the 1x iBind Flex solution that is provided by the manufacturer. And then also on the right, you can see the workflow. So the light blue parts in the process are the parts that are being done with the iBind device. Then let's have a look at the results. So here you can see the membrane images from traditional Western blot showing the different sized RBD proteins. So the sizes of the proteins that were generated were 35, 65, and 215 kilodaltons. And then as the housekeeping protein, we had GAPDH, and that sits at 42 kilodaltons. So because we wanted to assess the sensitivity of the method, we used different total protein amounts ranging from 10 to 1 micrograms. And as you can see, the RBD proteins were detected with all of these total protein amounts. Naturally, of course, the 10 microgram bands were the brightest, followed by the 2.5, and lastly the 1 microgram bands. One thing to note here is that the RBD2 and RBD3 bands seemed fainter compared to the RBD1 and RBD4, and this could be because they were larger and they didn't run optimally. And you can actually see that there is some smearing above the bands there. The smearing is not, however, visible in the 2.5 and 1 microgram lanes, even though the decrease in brightness is still there. In addition, there are these smaller transgene products visible in the RBD1 samples at 10 and 2.5 micrograms that might be either different glycosylation forms of the proteins or partial products. So you can see them under the main band there, kind of faintly, but they are there. And lastly, similarly to the RBD, the GAPDH was efficiently detected at 10 micrograms, with the bands being similar in all of the samples, indicating that the protein loading was equal. And then, of course, at both 2.5 and 1 microgram, the detected amounts were lower, but they were still detectable. Then for the iBind, so While the iBind Flex showed RBD bands of good quality in the lanes loaded with 10 and 2.5 micrograms of total protein, the bands were largely undetected at 1 micrograms. And this is even though we used the same exposure conditions or even increased the exposure times from a few seconds up to over 60 seconds. In addition, the smaller protein product that we saw in traditional Western blot in RBD1 at 10 and 2.5 micrograms is not anymore detected here at 2.5 micrograms. The GAPDH bands, however, were detected at all of the protein amounts, but either way, there's a clear difference in the sensitivity between traditional Western blot and iBind Flex, and it was consistently observed over repeated experiments. Next, we of course normalized the results, and here on the left you can see the representative Western blot images from the previous slides that I showed you, and there on the right the normalized results of 3 different independent experiments. So in traditional Western blot, the pattern of expression was the same at all different protein amounts used in the representative Western blot images, and in 2 out of 3 protein amounts in the mean data from the 3 different independent experiments. In contrast, the normalized data from the iBind Flex membranes did not really consistently repeat at the different protein concentrations in either the individual or the combined experiments. And this is most likely because of the lack of reliable detection of either RBD or GAPDH at the lower concentrations. However, we have previously used other antibodies that have performed well with iBind, so it is definitely antibody dependent, and this highlights the need to optimize all antibodies. So next, we move on to the simple Western Jess. So previously I mentioned the special plate that you pipette on, and here you can see an example of it, and the legend shows you where everything goes. So for the samples, we used concentrations of 100, 50, 25, or 12.5 nanograms per microliter, and from these 3 microliters was used per lane. The primary antibodies were of course the same that we used before, but now they were used at different dilutions, and 5 microliters was pipetted per lane onto the plate. The secondary antibodies and the ECL reagent were obtained from the anti-rabbit detection module for chemiluminescence, and we used a 12 to 230 kilodalton separation module that is also available from ProteinSimple Biotechnique, and then we used a replex module for sequential detection of the 2 primary rabbit antibodies, and this is basically analogous to stripping and then reblotting a membrane on traditional Western blotting. So looking at the results from the simple Western, while the RBD-containing proteins were clearly visible at all concentrations, the housekeeping protein GAPDH was very faint at both 25 and 12.5 nanograms per microliter, highlighting the difference between overexpression of a transgene and the expression of an endogenous gene. Here, it was also possible to detect some of the smaller-sized products of the transgenes that we saw previously in only RBD1, that are now also visible in the other RBD samples. On the left, what we're looking at now is a lane view image in the Compass for Simple Western Analysis software, but the results can be visualized also as an electropherogram from which peak heights and areas can be determined. And here on the right you see now examples of the RBD1, RBD2, and RBD4 with the solid line, and then with the dotted line you can see the GAPDH. Then for the quantitation and normalization of the simple Western results. So here on the left you can see the peak areas for RBD proteins, in the middle peak areas for GAPDH, and then on the right you see the normalized RBD from 3 independent experiments. So similarly to traditional Western blot, the patterns of peak areas from the RBD samples were consistent across all of the concentrations used, including the lowest concentration of 12.5 nanograms per microliter, and this then also mostly translated to the normalized expression patterns. However, whereas in both the traditional Western blot and iBind Flex, the higher molecular weight proteins RBD2 and 3 were detected at lower levels than in RBD1 and 4, here it was the opposite. So it looks like RBD2 and 3 are expressed at higher levels compared to RBD1 and 4. This could be possibly because of the suboptimal running of the large-sized RBD2 and 3 in the traditional Western blot, which resulted in the smearing that we saw on the membranes. One important thing to note is that for the simple Western, we only use 3 microliters of each sample, but actually the sample volume taken up into the capillaries is around 30 to 50 nanoliters, and this really highlights the sensitivity of Jess over the other methods tested here. Each of these methods, of course, have their advantages and disadvantages, and based on what is most suitable for you and your work, you choose the best option for yourself. If time is of the essence, some degree of automation is really beneficial. Whereas traditional Western blot can take anywhere from 1 to 3 days, iBind reduces the time of blocking, washing, and antibody incubations down to 2.5 hours, and it is incredibly easy and pleasant to use. In terms of Jess, it can complete the whole run in 3 to 5 hours, plus of course the time for sample prepping and pipetting and analysis. Traditional Western blot is, out of these 3 options, the most cost-effective, and many of the consumables are reusable, such as the gel equipment and the fact that membranes can be stripped and replotted. This is of course true for iBind as well, but of course in addition there are the device, consumables, and solutions to purchase. Then in Jess, there are the appliance and the consumables to purchase, which are expensive, but however, there is a lot of time saving that can perhaps offset the costs over time. As we saw based on the results, traditional Western blot is more sensitive than the iBind, at least with our tested antibodies, but it is still not perfect, which is why I've put sensitivity and reproducibility both in minuses and pluses. If we're talking about sensitivity and reproducibility alone, Jess really is the superior option, since with very small sample volumes and concentrations, the proteins were still detected in a reproducible manner. And this is especially beneficial if you have limited or precious samples like human samples or small animal tissues. Lastly, I want to thank everyone who made this project possible, especially Emma Koivulehto, who I share the first authorship of the paper with, and also the other authors, Kari Alitalo, Kalle Saksela, Mihail Karam, and Seppo Lehertola. I also want to thank Veli-Matti Leppänen for providing the plasmids, and Anne Martikainen and Maarit Mähönen for cell and plasmid work. And of course, I want to thank the Academy of Finland for providing the funding for this work. Thank you so much for your attention, and now I will hand it over for Rachel.
Thank you so much for the kind introduction. Hi, my name is Rachel Tallis, and I'm a field application scientist supporting Simple Western. Thank you for joining me today for an overview of total protein normalization strategies on Simple Western. Today, our speakers have touched on the ways that Simple Western supports quantitative research. With low sample utilization and an automated workflow, you can generate robust datasets that offer high precision in a 3-hour hands-off run. These benefits are enabled by our unique capillary technology, which is at the core of all of our Simple Western instrumentation. The run starts by loading the separation matrices into the capillaries, followed by the sample. A current is applied to separate proteins by their apparent molecular weight, like SDS page. Then, the proteins are bound to the inside of the capillary with our UV cross-linking technology. That allows for the removal of inert separation materials and the subsequent immunoprobing with the primary antibody of your choice and one of our ready-to-use secondary antibodies. The instrument images the signal from the immunoassay in each capillary and automatically analyzes the data in our Compass software. These automated steps happen over the course of a 3-hour runtime with no transfer or wash steps that could introduce additional variability into the assay. However, sample-to-sample comparisons of target proteins can still be skewed by differences in sample loading. Since accurate protein quantitation is essential to drawing meaningful conclusions about drug action, genetic differences, or disease progression, normalization to some standard is typically required. Normalization can be defined as a mathematical correction applied to account for differences in sample loading. The goal is to ensure that we are comparing biological differences between our samples, not experimental artifacts, to ensure that capillary-to-capillary comparisons are quantitative and meaningful. Two main methods are utilized for protein normalization: normalization to a housekeeping protein or normalization to total protein. Housekeeping proteins tend to be high abundance targets that are assumed to have uniform expression across samples. Some examples include GAPDH, beta-actin, or beta-tubulin. However, as this type of normalization relies on a single reference point, it is vulnerable to variation due to experimental conditions or treatment. Normalization to total protein is an increasingly popular method that captures the entire protein content of the capillary. Rather than relying on a single protein, total protein utilizes multiple reference standards, the breadth of the proteins within the capillary, which minimizes the impact of experimental treatments. Because of the susceptibility of housekeeping proteins to variation, many publications now require total protein data alongside immunoassay readouts. Bio-Techne offers 3 approaches to total protein normalization on Simple Western, each with its own distinct advantages. Depending on your sample type or required range, one method may be better suited than another. Having multiple options ensures that you can select the most appropriate approach for your experiment, making your Simple Western instrument more versatile and adaptable to different workflows. The first module I'd like to introduce is our total protein normalization module with chemi-detection. In this assay, your samples are loaded and separated in the capillaries. Once the sample is bound, we proceed with an immunoprobe to detect and quantitate your target of interest. Then, we utilize our replex reagent to strip the antibodies so the sample can be labeled with our biotin-based total protein labeling reagent. Signal is detected with a strep-davidin HRP probe and captured in the chemiluminescent channel. This module is exceedingly flexible and sensitive. It has a broad dynamic range that works for a variety of applications, from normalization of phosphototal detection to impurity analysis with purified material. It can be used with Abby, Jess, and Leo. The second option I'd like to introduce is total protein normalization with the NIR channel. This protocol utilizes the same robust chemistry as our TPN with chemi-detection, but expands the dynamic range of the assay by using strep-davidin NIR in conjunction with the biotin labeling reagent. This option is also compatible with replex, utilizing the second probe of a 2-step probing cycle. The TPN with NIR detection protocol has been tested on whole cell lysates, brain tissue, and musculoskeletal tissue homogenates. It's well suited for analyzing high concentration samples in conjunction with low expression targets. This module is compatible with our fluorescent-capable system, Jess, and will be compatible with the upcoming Leo fluorescence release. The third option is total protein normalization using the protein normalization or PN channel. This method uses a fluorescent dye that binds to all primary amines and is measured in a dedicated fluorescence channel, the PN channel. It provides a different detection range compared to the first 2 approaches and is fully compatible with our fluorescence-capable instruments. Unlike methods that use the replex module, total protein detection in the PN channel occurs before immunoprobing. This approach also supports multiplexing with any of the other 3 channels for a flexible workflow. Let's take a look at how total protein normalization is visualized in our Compass software. On the left, we have our lane view. This is an image based on our quantitative electropherograms that is modeled on traditional Western readouts. These samples were titrated, and we can appreciate how the blue total protein stain decreases in intensity with each dilution across the lanes. Next, we have an overlay of the total protein as a percentage. This is represented as a blue dot over the immunoassay, or the black bands. In this case, we can see the impact of each dilution as a percentage on the total protein dot. The signal decreases from 100%, where the protein load was the highest, to 50 and 25% and so on through the dilution series. That same decrease is reflected in the immunoassay bands as well. On the very right, we have the electropherogram views, which offer the most detail into your data. Compared to the lane views on the left, the graph views give insight into the peak area used for quantitation and provide more depth into the protein distribution throughout the capillary. These graphs I've chosen both show overlays of the total protein traces in blue. The top example shows the immunoassay detected in the chemiluminescence channel in black, and the bottom one shows the NIR and IR signals overlaid with the protein normalization in the PN channel. Corrected peak areas with normalization applied are displayed directly in the Compass software, ready to be copied and pasted and exported for analysis, presentations, or publications. Compass also offers a variety of advanced tools to ensure consistent and robust analysis. Here is a dataset that illustrates what an assay can look like with total protein in the chemiluminescence channel with immunoassay multiplexing. In this experiment, we were analyzing phosphorylation levels of AKT and MCF7 cell lysate. Samples were either untreated or treated with human insulin-like growth factor 1. HIGF-1 induces phosphorylation and activation of AKT. Using 2 antibodies that were specific to different phosphorylation sites, we were able to track the impact of the treatment on different samples. On the left, we have our lane view with the pan-AKT and phospho-AKT probes multiplexed in the NIR and chemiluminescent channels respectively. Below the immunoassay bands is the chemiluminescent total protein detection in blue. In the center and on the right, we have the graph views. This gives a more detailed look into the magnitude of difference in the phospho-AKT expression between the different samples. The concurrent total protein detection assures us that these differences are a reflection of the treatment and not due to poor loading or technical error. Now that we've introduced the different total protein normalization options and discussed what the data looks like, let's take a look at total protein normalization performance. Here, I have the description of an experiment that our application science team ran using our Leo system. HeLa lysate was run in various concentrations using a 2-step replex assay, with 1433 gamma in probe 1 and total protein with chemi detection in probe 2. The assay layout here shows the biotinylated ladder in capillary 1 and 24 capillaries of HeLa lysate. All rows were resampled for identical run conditions across 4 Leo cartridges for a total of 96 samples per run. In the first experiment, the lysate was run at the same concentration across 96 capillaries. At the top, we have our virtual lane view, which gives us a visual indication of the consistency of total protein detection. Below, we can appreciate the total protein detection graphed by peak area. The percent CVs are broken up by cartridge and range from 3.3 to 5.8%. That accounts for a cumulative percent CV of 6.6, which is consistent with the precision of simple Western immunoassays. With precision established, the next step was to demonstrate accurate normalization. We did this by running a titration series and recovering the target protein value using total protein normalization. With replex, endogenous 1433 protein expression was measured in samples ranging from 0.04 to 0.16 milligrams per milliliter. Looking at the lane view on the left, we can see the impact of the titration series on the black immunoassay bands, as well as on the blue total protein bands. On the right, we can see that the peak areas of 1433 reflect the titration of the sample that we observed in the lane view, resulting in an intra-run percent CV close to 50%. However, when protein normalization is applied, the area is recovered, bringing the intra-run percent CV to 6.7 and 7.5 on these 2 instruments, consistent with our precision data in the last slide. I've just shared some of our in-house data demonstrating the reliability and robustness of this method. Beyond that, simple Western total protein normalization is a proven solution used in more than 200 publications. It's driving advances in signaling pathway analysis, therapeutic response studies, and transgene expression validation, showing broad impact across a diverse range of research areas. What makes TPN powerful is that it carries forward all of the advantages you already know from simple Western: low sample requirements, shorter runtimes, and a hands-off workflow, while applying them directly to protein normalization. You also get the flexibility to multiplex with immunoassays in the same run, and the robustness of a protocol that isn't dependent on a single epitope, reducing variability. And because sensitivity is maintained down to the nanogram level, you can be confident that you're capturing reliable, publication-ready data every time. Thank you so much for your time and attention today. I hope I was able to share a little bit more about the vast capabilities of Simple Western. Next, I'd like to turn the discussion back over to our moderator.